It's that time of year. ACR Convergence 2024 is nearly here. Join us in Washington, D.C. from November 14th to the 19th. Meet up with colleagues from around the world. Enjoy thought-provoking sessions, listen to groundbreaking science, and join us for the Knowledge Ball, Thieves Market, and the Image Competition. Register today at rheumatology.org. ACR Convergence 2024, where rheumatology meets. Hello, and welcome to ACR Journals on Air podcast. I'm your host, Vicky Shamagam. When I was in medical school, I vividly remember opening up the Immunology textbook on the first day of class and reading a quote in the preface. Immunology, it read, is the invention of the devil, who is making it up as he goes along because he doesn't understand this stuff either. Well, it turns out the devil is in the details. The manuscript we will discuss today is unique interplay between anti-nuclear antibodies and nuclear molecules in the pathogenesis of systemic lupus erythematosus. It was published in Arthritis and Rheumatology, and a link to the manuscript and disclosures are in our show notes from today. It is a joy to be joined by the author of this review and a devilishly good writer, Dr. David Pisetsky. Dr. Pisetsky is a professor of medicine and integrative immunology at Duke University. He's recipient of the ACR Presidential Gold Medal and has held many leadership positions at Duke University Medical Center. He is both a clinician and a well-respected immunologist, having dedicated much of his career to studying the pathogenesis of lupus. Welcome to the show, David.
Okay, thank you very much. I'm very glad to be here.
So I want to talk about antibodies. In my ancient textbook, I learned the story of the LE cell and how anti-nuclear antibodies, or ANA, were first discovered. You cover this in the beginning of your article, but some of our dedicated followers may not be familiar with this story. So I wonder whether you could share it for our listeners.
It's a very interesting story. It goes back into the late 1940s. The story, as I learned it, concerned the performance of a bone marrow biopsy of a patient with lupus. The clinician who was performing the biopsy took the tube and got delayed on the way to his laboratory to examine the specimen after a right stain. After the delay ensued, he went over to the laboratory, stained the aspirate, and then saw cells that he had never seen before. And this turned out to be the LE cell, or the lupus cell. So it was quite fortuitous, a delay in the examination of that specimen, and then the discovery of this new cell type. As it turns out, that new cell type really has identified the theme of lupus that really persists until today. So the interesting part about it is a nucleus gets outside the cell. It's phagocytized or opsonized first by antibody, probably complement, then taken up into another cell. And what you see is a phagocytic cell engulfing the nucleus. And each element of that process remains important. The release of a nucleus from one cell, the binding of anti-nuclear antibodies, phagocytic uptake.
Yeah, so that's kind of a good lead into the whole premise of your article. You walk us through the various different autoantibodies in lupus. I wondered if you could also talk to us a little bit about anti-DNA antibodies, because as we all know, DNA is usually inside the nucleus. How does it get out for people to develop antibodies to it?
Yes. So, DNA is the essential model of heredity. It's always thought of as a nuclear molecule as opposed to other locations, and it's most certainly in the nucleus. But we should also remember it's also in the cytoplasm of cells because mitochondria have their own DNA. And that's cytoplasmic as opposed to chromosomal DNA, that's nucleus. But molecules get outside of cells. The major time of this is during cell death, when DNA and other molecules either get released for cells or just passively leave cells as the cell disintegrates, and then it can enter into the blood or the local tissues. You should remember that the translocation molecules from the inside to the outside of the cells occurs all the time. And some of our most useful biomarkers in all of medicine are internal molecules that have gotten out of the cell and get into the blood. Classic example would be LDH, lactic dehydrogenase, which is a very good marker of cell death and cell injury, which we rely on clinically. SGOP, SGPT, CPK, and myositis. These are all internal molecules that leave the cell. And nucleus DNA is just one of them. Now, DNA has special properties because it can become immunologically active. So in addition to being a molecule of heredity, it's also a signaling molecule. And the capacity for signal changes when it goes from inside to outside the cell. Now, during infection, there are also internal sources of DNA that can trigger these responses. And viruses, especially if it's a DNA virus, is releasing DNA into the cytoplasm. So DNA has locations, I shall say, all over. And I think one of the important advances in the field is to recognize that DNA, like other molecules, moves around and in other locations has different activities.
Yeah, that's really important to understand because damage in lupus often occurs through immune complex formation. And as you rightly point out, DNA is typically inside the nucleus of cells. The antibodies are circulating in the bloodstream, but it's the presence of DNA outside of the cell that then allows that immune complex to form. Can you kind of tell us more about how that happens?
Now, the classic and best studied example of immune complexes in lupus and certainly all of medicine is DNA, anti-DNA immune complexes. Now, as I say, DNA is ordinarily in the blood, but during inflammation, cell injury, other traumatic events, the amount of DNA rises and is readily measurable. Can get quite significant with infection, cell injury, following with cancer, following chemotherapy, anything that damages the cell. The other process to really consider is a process called netosis. Nets are products of neutrophils. They're called neutrophil extracellular traps. It's a complex problem that does involve cell death called netosis. And that is a physiological event that elaborates these extracellular DNA. And in this case, their function is to trap bacteria. So there are many ways that DNA gets into the blood. Now, as you indicate, the important aspect here is the formation of antibodies. And lupus, to a certain extent, is the only place where there are antibodies to DNA. And in this case, I'm going to be talking about double-stranded DNA in the B conformation. That's the classic Watson-Crick double helix. Just about the only place where those antibodies occur is in lupus. What's not clear is whether they arise specifically because the amount of DNA is in the blood has increased or whether there are other processes. Once antibody meets antigen, immune complexes are formed. They have two major actions. One is to deposit in the kidney and incite inflammation and lead to renal damage. And the other process, more recently recognized, is to stimulate cells of the innate immune system, such as the so-called plasmacytoid dendritic cells. In this case, the immune complex basically transfects the DNA into the cell. Inside the cell, there are cytoplasmic sensors to which this DNA binds, and that triggers an immune response. So we have a couple of events. DNA goes from the inside to the outside of the cell to form the immune complex, and then it goes back inside another cell, and there in the cytoplasm, it stimulates inflammation. So important thing here, there are at least two actions of anti-DNA, DNA immune complexes. One is nephritis, the other is cytokine stimulation. And this all harkens back to that LE cell phenomena, which is the uptake of a nucleus into another cell, and what's now recognized that's a signaling event.
Yeah, that's an amazing explanation. Take us back kind of one step before that and walk us through the different types of cell death, because that's also important in how the immune system sees these antigens.
All right. Well, classically, there were two forms of cell death that were recognized, and those were really an oversimplification. The two were apoptosis, the so-called programmed cell death, and the other was necrosis or traumatic cell death. So apoptosis is a physiological process. It occurs in many settings in which a cell is induced to die, frequently for a physiological reason. It's an enzymatically mediated process by enzymes called caspases, and it leads to the systematic disassembly of the cell as the cell dies. That process should be non-inflammatory. And one of the reasons it's non-inflammatory is so many cellular components are degraded, and therefore they're immunologically inactive. Now, as a counterpoise, there was so-called necrosis or traumatic cell death, which arises from physical or chemical damage that's not enzymatically mediated. Basically, the insides of the cell just spill out as the cellular membranes break down, and that historically was thought to be an inflammatory event. But this is really an oversimplification. It turns out there are multiple forms of cell death. There's apoptosis. There's a process called necroptosis, which is also enzymatically mediated. The outcome is different. There's netosis that I mentioned. There's so-called pyroptosis, which is an inflammatory form of cell death of certain types. Ferroptosis, which is another form of cell death in which iron atoms have an important event, and so on. Some of these are in vitro phenomena in systems that are, designed to allow a focus on death processes. Exactly how it takes place in vivo is still not clear. But it may be an oversimplification to say that one form of cell death, apoptosis, is non-inflammatory, and another form of cell death, necrosis, is inflammatory. Remember, one of our important mediators in rheumatology and immunology is TNF, tumor necrosis factor, and that's a cytokine that induces cell death. It's called necrosis. It's probably necroptosis. So many forms of cell death have physiological functions, but it can be pathological as well.
Yeah, really, really helpful. Now, there are some genetic conditions in which some of these enzymes that break down the cell, so these nuclease enzymes, are either damaged or missing. What do we learn from some of those unique genetic abnormalities? Do people with those genetic issues develop lupus?
Actually, interesting, there are many so-called single gene models of autoimmunity, of single gene models of lupus. And many of them involve steps in the pathway that I described. Some of the interesting ones involve enzymes that degrade DNA. These are usually cytoplasmic sources. This can lead to the so-called Aicardi-Gouttiere syndrome, which is associated with increases in the production of interferon. There are single gene models that involve the enzymes that degrade DNA. There are two main nucleases in the blood. One is called DNase 1, and the other is called DNase 1-like 3. They're similar, but their target molecules are different. DNase 1 likes or prefers so-called naked DNA, that's DNA that's not associated with protein, whereas DNase 1-like 3 preference is for DNA in the form of nucleosomes. Now, nucleosomes are the form of DNA in the cell. It's DNA wrapped around histones, and that is most likely the form of DNA in the blood. DNase 1-like 3 is the enzyme that degrades that form of DNA. So genetic deficiencies of DNase 1 and DNase 1-like 3 have both been associated with lupus, and they're a single gene molecule. The protein C1q is a DNA binding protein. Genetic deficiencies of complement are also associated with lupus. So many of the single gene models really involve some aspect of clearance of DNA. So it's a major theme. Now, I should add that some of these systems are also involved in infectious disease and susceptibility to infection. So bacteria have their own DNA system. There's extracellular bacterial DNA, and genetic deficiencies of DNase 1 and DNase 1-like 3 certainly in animal models are associated with increased susceptibility to infection. And that's because DNA extracellular DNA has an important role in infection as well. Finally, I should add there are antibodies to DNase 1 and DNase 1 like 3 so that there can be acquired genetic deficiencies of these enzymes. So this all goes back to that model of too much DNA in the extracellular space, either because of too much cell death or inadequate clearance, and certainly part of the clearance systems of the DNase enzymes.
So really the magic of the immune system is sort of the fact that in many people it stays in this perfect equipoise. I'm also rather fascinated by preclinical autoimmunity and the fact that we know that there are some very special people who develop autoantibodies like ANA and others, but never progress to clinical disease. Why do we think that is? And what could be done to study that group of people?
This phenomenon of preclinical autoimmunity is something that's observed in many autoimmune diseases. It's not easy to find people with preclinical autoimmunity. In terms of lupus, the study that really opened up this field was performed a number of years ago by the Oklahoma group, who used a United States military resource, and the military blood is collected on members of the military every year. What the investigators did is use this repository, identified people with lupus, and went back and said, when can we first find autoantibodies? And it was between 5 and 10 years. And that's a minimum estimate. Those people did not have lupus at that time. They had autoantibodies, but not lupus. And some period of time goes on before that predisposition or preclinical autoimmunity gets converted over into clinical autoimmunity. This is also observed in diabetes. It's probably observed in other disease. Again, the studies that allow you to find preclinical autoimmunity are not easy because you have to have a way of identifying people years before they get the disease. But people with rheumatoid arthritis also show this. So what accounts for this discrepancy between the autoantibodies and clinical disease? One, you could say it's the antibodies. So although these antibodies bind nuclear molecules or target molecules, they don't have the right properties to form an immune complex. Not every anti-DNA forms an immune complex, and maybe it just takes time for the antibodies to undergo somatic mutation to get some that bind DNA to form a so-called pathogenic immune complex. The other possibility is there's not enough DNA around, and what's required is inflammation of some other cause or injury that leads to extracellular DNA to move that process along. So UV light, for example, induces cell injury and cell death. Maybe that's what's required. Solar injury triggers release of DNA. Now immune complexes form. They stimulate interferon production, and just things get worse from there. That may explain what happens in lupus. You probably have to invoke other explanations for diseases like diabetes or rheumatoid arthritis. And maybe it's just some other event that's required to trigger this. And people always are interested in the role of infection, is somehow creating a setting in which this preclinical autoimmunity goes over. There are genes that promote autoreactivity in general. There are probably gene systems that promote the transition from pre-autoimmunity to clinical autoimmunity.
Yeah, and that leads us into the nice next topic that you discuss in the manuscript, which is tolerance. What is tolerance and why does it go awry?
Tolerance is sort of a general term that's invoked to explain the lack of reactivity of the ordinary or the normal host to their own constituents. So the immune system has to be able to recognize so-called self, that's the ordinary constituents of the organism, from non-self, and that is foreign bacteria and viruses. So self-reactivity culminates in autoimmunity and autoreactivity, and that is a very deleterious process. So there are mechanisms that prevent the generation of either B cell or T cell reactivity to ordinary constituents. This is a complicated process. It involves central sites such as the thymus for T cells, bone marrows for B cells, it also involves the periphery. There are many steps in which tolerance takes place. In the simpler format, it's a lack of reactivity to self, but it can be mediated by multiple mechanisms, and the failure of each of those in turn could lead to autoimmunity. And probably in different diseases, the failure to establish or maintain tolerance varies. There are genetic models of failed tolerance as well. So this is a highly regulated process. There is interest in the idea that as time goes on, this process wanes, and that as individuals get older, there is a tendency to have more autoreactivity, but not necessarily clinical autoimmunity.
Well, now, since you brought up age, there is a particular B cell population that seems to be quite important in lupus, and it's known as, dare I say it, the age-associated B cells, or ABCs. What are ABCs, and why do we think they're essential in lupus?
ABCs are a B cell population that have been recognized in a number of settings, and I should also add, they are defined by various markers. So they go by different names. The term ABC, or age-associated B cell, is quite apt. One of the places where they were originally discovered is just of older individuals and older mice. So it's a specialized B cell subset. It's distinguished by a cell surface marker. It's also distinguished by the transcription factor T-bet. These cells also increase in autoimmune disease, and they are antigen-experienced cells. They probably arise in the periphery. There seems to be a particular role of the toll-like systems, toll-7 and toll-9, in their expansion. And since nuclear antigens can trigger the toll-like systems, that may be one of the reasons there's an association with lupus. These cells seem to be a source of autoantibody production as well. So it's a B cell subset. It occurs in, again, ordinary situations, infection, older individuals, arises in the periphery, specific patterns of activation and transcriptional regulation, and an important role of the toll-like receptors. There are other B cell populations that get designated as double negative cells, and that really just relays on the type of the cell surface markers that are used for their different definition. These are also probably overlapping or the same population of age-associated B cells. So it's a specialized population. Now, clearly in lupus, this is an autoantibody-mediated disease. These B cells are obviously very important. And the question is, are there ways of specific modulating particular B cell populations or, in fact, eliminating B cell populations? We got the start in that direction with rituximab. Now there's interest in CAR T cells as an approach to really eliminate B cells, in particular, those more pathogenic subpopulations.
Yeah, so, that's a sort of tantalizing story in and of itself, a whole nother podcast and a whole nother article for you. So now, anyone who's ever treated patients with lupus knows that complement levels drop when the disease is active and typically normalize when the disease is in remission. Can you tell us a little bit about complement and its importance in lupus?
Now, complement is, I must say, a system where I would apply the term devilish. That's truly one. The rest of immunology is okay. Complement, not okay. Complement is a system that has role in many aspects of the immune system as sort of an amplification system. There are three pathways. There's the classical pathway, there's an alternative pathway, and then there's the lectin pathway. These are all ways of activating the complement system to produce complement split products that are immunologically active and then promote immune activity in various kinds in host defense. Now, in lupus, the main focus has been the classical pathway. So immune complexes form, they activate complement. That leads to decreases in C3 and C4, but also increases in so-called complement split products, which are the proteolytic cleavage products from complement that are immunologically active. So in the simplest possible model, immune complexes form, complement is activated, The split products promote inflammation. Inflammation gets worse. That's the simplest possible. So complement levels will decrease as you activate complement. C3 and C4 will go down. As the immune complex of burden is lessened, there's less complement activation, less production of these pro-inflammatory mediators, complement levels go back up. So you can either measure complement, C3, C4, total hemolytic complement, CH50, or the complement activation products. And they are now done by what are called cell-bound complement activation products, CB caps. Those are really complement activation products that are covalently bound to cells, and that allows a more ready assay of their presence. The correlation is not perfect. There are probably mechanisms other than immune complex formation and complement activation, so that C3 and C4 do not always correlate with disease activity. And at the moment, interestingly, it seems to be primarily DNA, anti-DNA complexes that activate complement. The role of other ANA complexes in complement activation is less clear. So it's a very useful biomarker. C3 and C4 are not the same in terms of their biology. That's why we measure both. And in the activity measures, you get credit for either C3 or C4, but there's additional points if you have both, because the pathways are somewhat different.
Yeah, and then I've always wondered, David, everyone has dead and dying cells. Cells turn over constantly. Why don't more people develop autoimmunity?
Well, one should add that the production of anti-nuclear antibodies is enormously common in the population. Depending on the assay used, between 10 and 20% of otherwise healthy individuals have an anti-nuclear antibody of some kind. These are not classical ANAs of the kind you find in lupus, but they are there. So some people think that those antibodies may have a physiological role in handling this burden of dead and dying cells. So one should say there is evidence. I don't think this constitutes preclinical autoimmunity because we don't in general know the target antigens that otherwise healthy people antibodies bind to. There are clearance systems for dead and dying cells. Some of the cell death is, as I said, non-inflammatory in the case of apoptosis. There are clearance systems, the complement systems involve phagocytic cells. There are many systems that are involved in trying to remove the burden of dead and dying cells. But systems fail, and that may be one explanation. But as I said, ordinary people have circulating DNA in the blood. Dead and dying cells are hard to find, probably because they're cleared so rapidly. There are just a few examples when you can see it. So the clinical entity, leukocytoclastic vasculitis, is an example of a situation where you see the dead cells, and the term leukocytoclastic is leukocytes that have come apart, and you can see the remnants. So that's a situation where you can actually see them. Otherwise, they're hard to find because clearance is so rapidly.
Yeah, the other thing that's always puzzled me is when you see a patient in clinic who has lupus and you're taking that initial history, you'll often find a family history of either another family member with lupus or another family member with another autoimmune disease. What do we know about genetics in lupus? Because clearly genetics is only part of the story, but it is certainly a common thread that we see. What's the scoop?
Well, beyond the single gene models or single gene systems, genetics of lupus, like the genetics of other autoimmune, is very complicated. So at this point in time, between 100 and 200 different gene systems have been identified as contributing to lupus. In general, the major locus is the major histocompatibility complex, the HLA region in humans. That, in general, has the strongest association with autoimmune disease. The other gene systems have much lesser roles. HLA is itself complicated. It is involved in recognition of specific antigens, but it's also the region that encodes complement, it encodes cytokines. So one can imagine one inherits general tendencies to autoimmunity in a family. But given the way heredity works, outside of identical twins, everybody's different. So it's this association of different predisposing genes in an individual creates a predisposition, and then likely something happens. In my own work, I favor infection. Doesn't have to be a very specific kind of infection like EBV, Epstein-Barr virus. It may be more conventional infections. That triggers the tendency. In another family member, because the genes are similar but not identical, the combination may be a different autoimmune process. So in families, as you indicated, you find different autoimmune diseases. And I think that's because there are certain common genes that underlie autoimmunity, but other genes that detail the specific kind of autoreactivity that would be found, and then still other genes that determines that transition from preclinical to clinical autoimmunity.
So I do think we've probably moved beyond thinking of immunology of the devil. How has this improved immunologic understanding impacted our ability to treat people living with lupus and living with other autoimmune diseases?
Well, there's both treatment and then there is prevention. In diseases where there's a high frequency in families, the example here is type 1 diabetes. This has actually led to clinical trials to see if diabetes can be prevented. That effort is assisted by markers, autoantibodies of a certain kind in diabetes. Those can be followed in the phase of preclinical autoimmunity, and then efforts to use immunosuppression to prevent that transition of preclinical to clinical autoimmunity, or really to attenuate the damage from autoreactivity. So I think in diabetes, we're the furthest along because we can focus on particular patient population. Also, it occurs relatively early in life. For lupus, what this has led to is really a wide array of different therapies that target different aspects of the process. There are anti-inflammatory, there are immunosuppressive agents, and there are now new agents under development that affect particular cell population, particularly B cells, and there's cellular therapy. What we're not at in the stage of lupus is prevention, because it's not common, not that common. It's difficult to follow the patient population. Now, I would say every rheumatologist has seen a patient who's ANA positive, who has some type of clinical problem. Whether it's pain, whether it's arthritis, whether it's rash, or just symptomatic. And always the question is preclinical autoimmunity? Are these people at risk for going on? And is there anything that one could do to prevent that transition? In that setting, autoantibodies are very helpful to see if there are antibodies that are clearly associated with lupus that would allow early therapy or preventive therapy. The challenge here is that autoreactivity, or the ANA positivity, is so common in the population that it would be difficult to see all those patients in follow-up. So what would be needed are biomarkers. Autoantibodies are probably pretty good ones at this point. Complement determinations are useful, but other markers to say that this person is on the way to lupus in a way that preventative or secondary preventive therapy could be given.
Yeah, so maybe the promise of the future of immunology is that we'll actually have some biomarkers of risk of progression and potentially risk of flare that might actually help us tailor those therapies more accurately to the patients.
That's exactly right. These are not easy studies to do. And I think the experience in rheumatoid arthritis is probably a good example of trying to say what is preclinical autoimmunity as opposed to actual autoimmune inflammatory diseases. So what some people would say is arthralgias, in the absence of more objective signs of tenderness and swelling, would be more indicative of a preclinical or early disease state. In my mind, once there's arthralgia, to me, there's probably already established autoimmunity, but nevertheless, treatment is very, very worthwhile at that stage. So this distinction of preclinical to clinical autoimmunity is not easy, but clearly there are people who have autoreactive who don't go on to major clinical disease. So having markers of some kind to say that this person has a high likelihood. I think the other term that's now being used rather than, or in addition to clinical autoimmunity, is at risk. So individuals who have autoantibodies are at risk for prevention to progression to autoimmune disease, especially if it's expression of antibodies like anti-DNA or anti-nucleosome, SMR, and PROLA. They really are at risk for lupus. And ANA positivity alone is not as great a risk or not that much of a risk. It's the production of specific autoantibodies. But we don't know, because it's hard to do this kind of study, is if someone has autoantibody like anti-DNA, anti-SM, and so on, what their risk is. That we don't know precisely.
Yeah, and timeline also throws us off, right? We don't know how quickly, or the
we don't know
time varies in different people.
And I think it's not easy for patients. I think you have to put the patient in the equation. It's very hard to tell patients you're at risk for a disease unless you have a preventive strategy. If you have a preventive strategy, it's much easier to get patients to understand the situation and adhere. If you have a therapy that doesn't have too much in the way of side effects, like hydroxychloroquine, for example, then one could think of progression. But patients with these autoimmune diseases confront a lot of uncertainty, and they go from physician to physician, not infrequently, what do I have? And you hear stories of patients going years who are symptomatic, not clear if they have lupus or they don't have lupus. So this idea of preclinical or at-risk disease, I think, is very attractive, but I think you have to put the patient in the equation to figure out what the impact is. How often do you test? What triggers you now to go on to treat? And if preventive treatment doesn't relieve symptoms, it's much more difficult to get adherence.
Yeah, I totally agree. David, I want to thank you for sharing all of these insights and all of your knowledge about the advances in immunology in lupus. I love your articles because I always feel much cleverer after I've read them.
Thank you.
So during the next segment of the discussion, I want to give our listeners a chance to meet you, the person behind the paper. And I wondered if you'd be willing to share a little bit about your career. You completed your internship and residency at Yale University, and then, like many rheumatologists, you spent some time at the NIH. How did that come about, and who did you work with?
So just about a bit of history. So I went to medical school 67, 73. At that time, there was still a draft for the military in the United States. And physicians, like everybody else, had military service requirement. And for a physician, there were a number of ways that requirement could be fulfilled, one of which was service at the National Institute of Health as a member of the Public Health Service. And at that time, the Public Health Service was a branch of the Navy. So when I went to the NIH, I want to say, believe it or not, I was actually a commissioned officer in the Public Health Service. Now, for me, that worked out great. I'm a scientist. I wanted to go train. NIH is an outstanding place to train. I wanted to be in immunology, worked in a top immunology lab. But just by history, there was a draft, and doctors, like other people, were required to do military service. At the time I entered the NIH in 75, the military draft went away, and now one just elects to go to the NIH. It's outstanding training. I went to be a basic immunologist. I worked actually in the Cancer Institute. My clinical work was immunotherapy of malignant melanoma, but my lab work was fundamental immunology, learning about antibody response. I focused on antibody responses to a model antigen. Turned out to be a nuclease, but I got very interested in using what I had learned as a basic scientist, looking at genetics and mechanisms of immune response to apply to lupus. So even though I was in the Cancer Institute, my clinical interests were in rheumatology. So it was a great experience. It's outstanding training. I'd recommend it for people who are interested in being physician scientists.
Yeah, so you were ahead of the curve even back then. So you've now been at Duke for many, many years. What tips can you share about surviving in academic medicine?
One, I would really say for people who are thinking, take on a big challenge. I know that's not always easy advice. I think people get engaged if they see a big challenge. For me, it was to understand a disease, understand lupus. So I think it's have a problem that you really like and you're really engaged by, but also it should be big. So think big would be the first thing I would do. Get out, get top training. So I have a PhD plus a postdoc. You need to train, and it takes every bit as long to be a laboratory investigator as it is to be a clinician. So I spent 6 full years in the laboratory doing my own experiments. I think you need to do that to understand design of experiments, interpretation of data. Have good colleagues, learn how to collaborate, but also I would say learn how to be we always talk about mentorship, but I think what's also important is to be a mentee and to have a mentor who's a top scientist and learn what you can from that individual. So I think those were things. But I think what really is, to the extent there's a secret, is having a problem that engages you and challenges you. I've been working on antibodies DNA basically for 45 years. I still find them very interesting.
Mm-hmm. Still gets you up in the morning, which is very good to hear. I wondered if you could tell us a little bit about your writing practice. You've written over 500 scientific papers, many textbooks, and an excellent book commemorating 75 years of the American College of Rheumatology. How do you write? Are you someone who needs a silent room and a computer, or are you more of a longhand scribbler and get in the zone?
I write different ways depending what I write, I should say. And I write different kinds. As you know, when I was editor of the Rheumatologist, I wrote essays or narratives. Scientifically, I write conventionally. I actually went to writing school. I should just add. I got started in what I'll call non-scientific writing. I was asked by my department chair to write a book, which I did, called the Duke University Book of Arthritis, which is a book directed to patients. And I went through that process trying to learn, how to write in a different way than you would scientifically. So I took a number of writing courses, which I think helpful. As I said, I write differently. When I was an editor of the Rheumatologist, a lot of what I wrote was longhand, and I did it on airplanes. I traveled at that point, and it was a good way to spend the time. If I write science, I write in my office silently. I used to be a fiction writer. I would write with music. So it really depended on what I was writing. I would never try to write science with music. And correspondingly, I'd never write fiction silently.
Oh, that's really interesting.
So I find that very interesting. But what I think when I was added to the Rheumatologist, a lot of what I wrote about was travel. If you look back, it was travel and sports and then rheumatology. So I think travel was a setting where you have time to spend, especially if you're on an airplane. And pen and paper was just a very good way to do it.
Yeah. And writing for fun is obviously something you really enjoy. As I was researching for this podcast, I came across a piece you'd written some years ago for Scientific American called Science at the Baseball Plate, where you wrote what I can only describe as a parody of a baseball game in the style of a scientific article. It definitely made me chuckle. And then it made me wonder, should we make more space for fun in science writing in our journals?
I'm going to say absolutely. So just by background, I was the Wallace Epstein visiting professor at University of California, San Francisco. Wallace Epstein was a great investigator. He actually started as a more conventional immunologist and then went off into health policy. Outstanding individual. And there was a lectureship in his name. And they asked me to talk about writing. So I actually wrote 3 separate pieces for that visiting professorship. That was at San Francisco. It was the time that the baseball giants were playing the Yankees. I'm a Yankees fan. And I just took it upon myself. How would a scientist write about the World Series? And that led to that article. I wrote one that was published in ACNR about translation about current style of writing, how long articles are. It was called From Treatises to Tweets, The Extremes. And then I wrote another article, which I'm still working on almost 10 years later, about describing my life as a scientist. So 3 articles came out of that. Could we write better? Absolutely. I mean, what baseball plates indicated is sort of this stultifying and stilted way we write as scientists. For example, in the passive voice, we all know who's doing the experiments. We are. I am. Why can't I use it in the first person? I did this experiment, or I thought this, or I thought that. So I think we could just make life a lot better by simplifying it. Articles should be shorter. They should be more direct. They should be more accessible. We're in an era of big data, which I'm going to say, unfortunately, because it doesn't lend well to simple writing. There's too much data. I'm becoming increasingly the belief that review articles, like the kind we're talking about, have great value to synthesize a field in accessible format. Not all the details. There are too many details. I like the process of writing. What I tell my trainees is science is really about reading and writing. Experiments are just a way of collecting those two. A lot of people say, I want to get into the lab. And I say, no, don't go into the lab until you know what you're working on, and go read about it first. People don't like that. But that's the reality. But you can't be a scientist unless you can write. You know, if you can remember, I don't know what your schooling was like. I never, in high school, college, did a term paper. I did a problem set. So I was never taught how do you write about science. And I think that's a mistake. I would say, you know, we don't need all those details, but we need how to convey the information. So I think there really is an opportunity for clearer and better writing.
I completely agree with you. And it's actually something we don't truly teach in medical school or in our training of physicians, is how to write well, both for communicating your science and communicating your science to the general public. We don't do a great job of that.
We actually write a lot because we write histories and physicals. So every patient encounter really leads to a great number of words, but it's formulaic.
Mm-hmm.
So when I teach clinical, at least for a long time, patients, a student would say, the patient looks older or younger than their stated age. It was not an uncommon. And I would say, why? You know, tell me what you saw that led to that conclusion. And that will never show up in the medical record.
Mm-hmm.
What will show up is they look younger than their stated age or older, or they look chronically ill, or they look, you know, is what led you to that conclusion? So I think what happens on the clinical side is we never ask people to really put down their observations in a coherent way. We just put down the formula. And as a writer, what I think you should really do is go beyond the formula and tell me what you really saw. So I think my science writing has been influenced by my non-science writing, and to a certain extent, the other which way around. But I think the non-science writing emphasizes description and accessibility so people know what you're doing. So I think you could do that in science readily, if you were allowed to. Journal editors sometimes say no.
Yes, and I do love your writing and the fact that you can actually take a reader along the narrative arc of the story, even when it's very complex immunology. So I appreciate that some of that may well have come from your training as a fiction writer and not as a science writer, but I definitely appreciate that skill.
Right. I mean, basically a science article is a short story. It's not a novel, it's a short story. And there should be an emphasis on making it interesting and want the reader to go on.
Yeah.
And you want to tell a story. Sometimes you have to give it structure, and a lot of this is post hoc because science changes. But what I think is so limiting is that you're not able to express your feelings about your own work. You can't say, boy, this was really interesting.
Yes.
You can't do that.
You can't tell them the eureka moment. You have to
can't
try and let them find it.
You have to try to do so. That's why you have to write in a different way, is to make it seem exciting. Because just saying, wow, this was really interesting. Uh-huh. I mean, there's a eureka quality of science, but if you ever put in a journal article, eureka, I'm telling you, someone would tell you to take it out. Even though
For
that's what
sure.
scientists would think.
Yes, absolutely. So I really want to thank you, David, for joining us today and for sharing all your expertise about lupus, antibodies, and a little bit of the lighter side of life.
Thank you very much. I've really enjoyed it and appreciate it.
My thanks today also go to writers, especially scientific writers who can take something complex and make it understandable with the power of words. Without you, immunology would still be devilishly difficult. Thank you all for listening to this podcast. ACR Journals on Air is a production of the American College of Rheumatology. Please follow at AC Rheum on social media. Email us at podcast@rheumatology.org and rate and subscribe to our podcast at acrjournalsonair.org or wherever you get your podcasts.